Good afternoon and welcome. We've got another episode of Fertility Talk, and today we're talking to Neville Moodley. And Neville is an embryologist or senior embryologist at Genesis Reproductive Centre. I'm going to have him introduce himself. But we are talking today about the topic today we're talking about is the ICSI procedure step by step from sperm collection to embryo transfer. So thank you, Neville, for joining us for taking the time out because I know you're a very, very busy guy. So thank you for taking time out to join us today and discuss this with us. Neville, please tell everybody what is it that you do? What is it that you do at Genesis?
Thank you so much for having me here on Fertility Solutions. Yeah, as you said, my name is Neville Moodley. I'm the lab director and senior embryologist at Genesis Reproductive Centre. Besides being the lab director, I'm also the operations manager at Genesis Reproductive Centre. So we manage the affairs of the clinic, especially within Genesis. So it's a very exciting portfolio that we have because it's a clinical aspect where of the laboratory and clinical procedures that are done at Genesis and also the operations as well.
Okay, thank you very much, Neville. So we're going to get into the questions. We've only got 5 questions for you today. Like I said, we're going to talk about ICSI. We're going to get into our first question is, what are the steps involved in sperm collection for ICSI? And are there any specific preparations or requirements for the male partner?
So I think to go forward first, we just need to have an understanding of what the process of ICSI is. ICSI is an acronym for intracytoplasmic sperm injection. So it basically into cytoplasm, that is the cytoplasm of the egg. Cytoplasmic sperm is the sperm itself, and injection. So it's basically injecting the sperm into the egg. So it is a process where a single sperm is injected into the egg, and then we put it into in culture then to see fertilization and embryo development. For the sperm selection process, sorry, you said that it was a sperm retrieval collection process.
Yes.
So the sperm is the ideal aspects for sperm collection is this is with regards to the male aspect. You need to have an abstinence of between 2 to 7 days. The ideal number of days would have been between 3 and 4 days, but it's between 2 and 7 days. The actual collection process is done by masturbation, where a patient produces a sample via masturbation, and we do not like where it is any we actually avoid patients from having any intimacy, that is, any oral intimacy, etc., because the fact is that that will show a contamination in the sample. When patients have difficulty in producing a sample because of cultural beliefs and also religious beliefs, then we also provide a special condom. And why we say special condom? Because normal condoms contain spermicide. Spermicide is something that kills sperm. Hence, condoms are used for contraception. But these condoms that we have for fertility clinics do not have spermicide. So they're devoid of spermicide, and we can actually work with the samples from there. So the patient can use a special condom, have intimacy at the nearest B&B or whatever the case is, and then bring the sample to us within 45 minutes, and then we can start to process the sample from there. On patients that have more difficulty, let's just say for reasons of like they're not available to produce samples on the day of the aspiration, then we can also freeze sperm in advance and keep the samples. So, for example, let's say the husband is working or the partner is working and cannot make it on that day. He's got to go overseas or is in another province or whatever the case is. Then we know we have the sperm that has been frozen as a backup for it. And then the last part would be that the patient is using donor sperm, and the donor sperm could be frozen donor sperm as well. With the sperm processing part of it, we can either do a density gradient centrifugation, that is where we would separate the sperm to see get good moving sperm and good morphologically moving sperm via density gradient centrifugation, where we spin the sperm into, we actually layer sperm onto colloidal silica particles that have different densities. And what happens is that then now it's like a filtration process. Sperm has, when your semen is the sample that has been given by a male. In the semen, it has four parts, which is one, the sperm, which is the most important part, and then it has secretions of three accessory glands. That's a prostate gland, the seminal vesicle gland, and the urethral gland. So you're going to find that you're going to have living sperm and you're going to have dead sperm because sperm have a half-life. They don't last completely all the time. They're not alive all the time. Then what will happen to the sperm? You're going to find in a fresh ejaculate, you're going to find some dead sperm, some twitching sperm, and you can see some moving sperm or have good forward progression. Our job is to filter the sperm so we can get the best sperm, and from that we will be able to use the best sperm either via the filtration method of either using a density gradient centrifugation, also called a DGC, or a swim-up method as well, in which we can get the best sperm to inject the sperm.
Okay, thank you very much. Thank you very much, Neville. Our second question is, how is a single sperm selected and injected into the egg during the ICSI process, and what factors influence the selection of the sperm?
So I think what's important is that when we prepare our dishes for injection, we prepare them with little droplets, like 20 microliter droplets, and we put the egg into the droplet in order for us to inject it. But we also make another line, and that line has got a thing called PVP. Now, PVP, when we process the sperm, the forward progression of the sperm is activated. And what happens is the sperm starts to swim very quickly. Our job is to immobilize the sperm firstly. So the only way to do that is that we put the sperm into a PVP drop. And the PVP, what it does is it slowers the movement of the sperm. So it doesn't hinder any aspect of the sperm or damages it or anything of that. So the only thing that it's doing is it's slowing the movement down. It makes it a bit easier for us in order for us to catch the sperm. So when we see the sperm that is swimming, the factors that we look for is firstly we're going to look at the shape of the sperm. I think one of the important things is that we're looking at the shape of the sperm, shape of the head, which is a very important thing. So we look at, it's called morphology. And the morphology we're looking is for an oval shaped head with at least one third of it, the acrosomal region, and two thirds of the post acrosomal region, which we can see on the head. We also look for stuff like the neck insertion. How is the neck inserted into the sperm? Is it inserted directly in the middle of the sperm, or is it on the side, or whatever the case is? So it also shows us, that's not a good sperm for us to take. And one of the other important things and factors that we look at is how the sperm is swimming. We want to try and get the ones that have good forward progression, that have good, like a torpedo type of movement that is swimming. And when you see the sperm swimming, because it's a 3D, you see like the head, it's like a torpedo. It moves like this. And what it is, you can see it changes shape as it's moving. But it's actually not changing shape. It's just that it's moving, swimming around and going through it. It looks like it's changing the shape. When we find those sperm that we actually like, we will catch the sperm. Now, people always ask us, how do you catch the sperm? So it's very basic. We have these micro manipulators that we are working with. One is a holding needle that we have, and the other one is an injection needle. The injection needle, what we do is that we find the sperm that we want, and then we swipe the tail. Now we swipe the tail so we can immobilize it. Please note, we're not killing the sperm. We are just immobilizing the sperm. When we immobilize, why do we immobilize? We immobilize it because when the sperm normally goes to the egg, there's a series of events that happens. One of the first thing that happens is called the acrosome reaction, because the sperm head normally has a thing called acrosin, which is an enzyme that allows it to digest into the glycoprotein layer of the egg so that it can enter into the egg. But then once it's inside the egg, then there's another process called capacitation that occurs, and that is the process where we are trying to mimic the same things that the sperm would naturally do. So we swipe the tail so we can activate the process of capacitation of the sperm, which is important because it's like the sperm in layman's terms, telling the egg, "Listen, I'm here. I'm ready to meet you, etc. Let's do our thing."
Okay. Thank you very much, Neville. Next question is, what happens after the egg is fertilized through ICSI, and how is the embryo monitored and cultured before transfer?
So I think what's important to understand is that firstly, when as soon as we inject the sperm into the egg, it's not fertilized. We are just producing this, introducing the sperm into the egg. When we introduce the sperm into the egg, we also have to understand that the genetics are involved in this whole thing. A mature egg would have one polar body. And people ask, what is a polar body? So you see an egg with a body, a round body that is called a polar body. That shows us that the egg is a mature egg and we can inject that egg. So we can only work with mature eggs, metaphase 2 eggs, those are called, right? So when we inject the sperm into the egg, then we leave the sperm there. The sperm now, I'm just want to explain this in very layman terms
Yeah,
because I don't want to
please.
make it too hectic. So it's like this: the sperm enters there. It's like me meeting a person for the first time. Hi, how are you? This is who I am. So then what happens is the sperm's head swells and it shows its DNA to the egg. So when it shows, that's called a pronuclei. When it shows its DNA to it, and you can see the pronuclei, that has 23 chromosomes. Now, the egg generally has 46 chromosomes. Now, a normal human being, like you and I don't know why I said normal human being, but a human being has 46 chromosomes. That's the normal complement of your DNA that you should be having, 46 chromosomes and a pair of sex chromosomes. However, now when the egg is already have 46 chromosomes, but when the sperm enters now, it's also bringing 23 chromosomes as well. So when it brings in the 23 chromosomes, you can't have 46 plus 23 because it's too much of DNA. So in reply to the sperm saying, hi, here am I, my name is so and shows its DNA, the egg in response will also say, oh, okay, this is who am I. So she will also show her DNA, 23 chromosomes. So it would be 23 chromosomes from her, 23 chromosomes from the sperm. And then the other 23 chromosomes that the egg had, because it was full complement of 46, is extruded out into the egg as a second polar body. So when people go through fertility clinic, they will always fertility treatment. They will always hear the embryologist say, "No, we had two pronuclei and two polar bodies. That's how we know it was normal fertilization." In order for us to show normal fertilization, there must be two pronuclei shown. One from the sperm to say, "This is my DNA, 23 chromosomes." The egg will say, "This is my DNA, 23 chromosomes," and two polar bodies because the second polar body means that the second polar body was extruded to show that it was normal fertilization. From there, then we would then that's the first place to see how fertilization and to see if the fertilization is normal or abnormal fertilization. And from there, we will then start the culturing method. The culturing, there's different types of culturing methods. You have one which is first called a sequential culturing. Sequential culturing means that the embryologist will prepare media on different days, and then the embryos will be transferred on different days to different types of media. And generally, the changes take place on day one, day three, and you prepare for day five for either embryo transfer or freeze. Then you have a one step, a single media culturing. Single media culturing is that you use one media, you do the fertilization, and then you transfer it into a media that is used for the entire 5 days of the culture. So the difference between sequential and one step and single step culture is that where sequential, you change it on different days. You're putting it into new media on different days. That's either day one, day three, and day five. But in single step culture, you are putting it in day one, or you've done the fertilization, you change it on day one, and then you leave it completely until day five. So more different labs work differently, and then the last, it's not really a culturing technique, but it's a way they use time lapse. Time lapse is where they would put the media either generally normally is a single culture because with time lapse you don't want to open the incubator all the time, and then you can actually see what is happening with the culturing.
Thank you very much. Our second last question is, can you explain the embryo transfer process, including how the best embryo is chosen and what the patient should expect during this procedure?
Yeah, so I think what's firstly, the embryo transfer process is quite um, it's not really a difficult process to do. The doctors can have sometimes difficulty because the patient's uterus may be lying in a different plane, and then you've heard doctors talk about retroverted and antiverted uteruses, and it just shows the different ways in which the uterus is being lying at that position. So embryo transfer is very similar to how people do a pap smear. So it's when I say pap smear, it's the same. It's exactly the same procedure. Doctor puts in a speculum, he exposes the cervix, cleans it, and makes sure that there's no mucus plugs and so forth, and then passes the flexible catheter through the cervix. And they normally do it via an ultrasound guided transfer, so they can see exactly on the ultrasound where the catheter is going to, and they leave it at between 1 to 2 centimeters away from the fundus. And the fundus, so if you think about it, is like this here. You got the catheter going up, and then this is the base of the uterus. So it's left about 1 to 2 centimeters away from the fundus in the endometrium lining there, in order for implantation to occur. So it should be atraumatic in the sense that there should not be any blood or anything of that sort when the transfers are being done. So best transfers are one that which is atraumatic. But I mean, you do get patients that transfers can be difficult. But I mean, every patient reacts differently, and even patients that have sometimes difficult transfers still fall pregnant, and everything works out well for them. I think what's important is now how do we pick, that is just the embryo transfer technique. How do we pick the embryos? We work on a grading system. So I'm talking about the blastocyst now, which is generally produced on day 5 or day 6 of your culture. There are different grading systems, but we use the Gardner, or the most commonly used one is the Gardner grading system, and it's named after the one of the very senior and expert embryologist, David Gardner. So we use three criteria that we're looking for the grading. The first one is you look at the expansion of the blastocyst. How did it expand? Remember, if I can just take one step back before that, we have fertilization. Once we have fertilization, the sign of chance to show that the fertilization, the embryo, it's becoming an embryo, is it starts to divide to form an embryo, where to form 2 cells, 4 cells, 6 cells, 8 cells, etc. On day 4, it should be a morula that is more than 16 cells. And then on day 5, it forms this highly complex structure, which is called a blastocyst, which is more than 100 cells. And it has two distinct parts, which is called a trophectoderm and an inner cell mass. Now, the trophectoderm becomes the placenta, the inner cell mass becomes the actual baby. And the egg will hatch like normal, like how you get a normal egg that will hatch. The egg hatches out, the placenta, the trophectoderm comes out, it's still holding the inner cell mass. When it comes out, it will attach onto the endometrium, holding the inner cell mass. So remember, trophectoderm becomes placenta, inner cell mass becomes the actual baby. So we look at the expansion. The embryo needs to expand, 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 or the blastocyst needs to expand, and then it'll, because as it's expanding, it'll push the egg open, have a puncture site in which the egg will, the trophectoderm can come out through the zona pellucida. That's the outer structure of the egg. So we look at the expansion grade. We grade them from grade 1. So it's 1, 2, 3, 4, 5. Grade 1 will be an early blastocyst. The blastocele cavity will be less than half the volume of the embryo. Then we have grade 2, which is a blastocyst, where the blastocyst is equal or greater than half the volume of the embryo. Grade 3 would be when the blastocyst, it's a full blastocyst and it's expanded nicely, and the blastocele cavity, that is, you'll see that formation is completely fills the embryo. We have grade 4, which is an expanded blastocyst, which the volume is now larger than that of the early embryo. And the zona, remember that I spoke about that zona pellucida, which is the outer shell, will be thinning really, really nice and thin. And then grade 5 would be a hatching blastocyst, where you have the junction where it's penetrated. The egg is actually hatching through, and the trophectoderm like herniates through the zona. And then you have grade 6, which would be a hatch blastocyst. That is when it's completely out of that zona and it's hatched completely. Then that's the first one grading. So you will see either the 1 to 6, um, that the first one. Then you would see a second grading system, which is for the inner cell mass. The inner cell mass grading we look at is graded A, B, or C. A would be the inner cell mass is nicely. It's a large inner cell mass, which is tightly compacted properly. And then you see B, which is the inner cell mass is a bit smaller in size. There are a few more loose adherent cells, and it's less compacted, so it's a little bit less compacted. And then C is very few cells in which are visible, and it's not so large as A and B, and it may be very loose cells around and difficult to distinguish from the trophectoderm. Then the last one we look at is the trophectoderm grading. The trophectoderm grading is also A, B, and C. Now, trophectoderm should have a nice ridge-like appearance on the cohesive ridge-like appearance. So A would be there's many identical small cells that are forming tight knit of cells around it, and appears evenly. They call it scalloped, so it is even the ridges right through the blastocyst. B would be that it has fewer smaller cells, but you're still going to see the ridge-like. But sometimes you're just going to say um, continuous uneven layers. So you may have a ridge, and then straight, ridge, straight, so it's uneven layers. And then you get C. C is when there's very few cells of uneven sizes and it's forming loose structures all over the place. So you would see that they would grade the embryo. You would have heard like a 4AA, 5AA, you would have heard a
Yeah.
2BB, 3BB, or 3BC, 5BC. Those are the gradings.
Okay. Thank you very much. Our very last question for you is, what are the potential risks or complications associated with ICSI, and how do you manage these to ensure the best possible outcomes?
So I think ICSI is a very high complexity process, and there's IVF where you leave sperm with the egg for it to naturally find the egg, and to allow it naturally to fertilize with the egg. But with the ICSI, it's manually operated, where the operator, who is the embryologist, is physically injecting the sperm
Yeah.
into the egg. So in that way, we're already mitigating the part where you can get no fertilization, which which would have happened with IVF, which can happen. One of the things that can happen is that when the operator is actually injecting, if the egg's zona pellucida, which is the outer structure, or the oolemma, that is the inner membrane that holds the cytoplasm, is fragile or too thin, sometimes when you inject the egg it may not retain its shape, or may not retain, because remember we are injecting. It's a very small needle that is being injected into the egg. And once you inject it, normally the ulema goes onto the egg and forms like a funnel process to show that it's a good injection. But sometimes it can collapse onto the needle whilst when you inject the egg. And if you inject in such a way, sometimes you may create a larger puncture into the egg, which will cause the egg to degenerate as well. So that's one of the risks of it. It doesn't happen all of the time, but it's with skilled embryologists that you don't really get that all the time. But you do get degeneration of eggs that can happen as a reason of the egg being very brittle, and also the zonas being very thin as well. The other thing you can have is you can get abnormal fertilization, and this is where you can get a three pronuclei forming. And that is, the three pronuclei is not because we injected lots of sperm into the egg. It's because, remember I spoke about the egg has 46 chromosomes, the sperm has 23 chromosomes, but sometimes the egg doesn't extrude that second polar body out, and it
Yes.
still keeps it out and shows its DNA. So now you got 23 chromosomes, the egg is showing here, and 23 chromosomes she's showing here, and also the sperm is also giving its 23. So you're going to see three pronuclei forming because the second polar body has not been extruded. However, I mean, ICSI is one of those processes, which is supposed to better the chances of fertilization because of the operator that is injecting manually the sperm into the egg instead of us just putting sperm there and waiting to see if do
Yeah.
they buy, do they not buy, what is the problem here. So those are the per se risks and how we mitigate against it. But on the same token, I mean, these are much more highly complex and sophisticated technique.
Okay, thank you very much, Neville. We really appreciate your time today to go through with us. And I love the detail that you gave with everything. And I always feel like I'm learning quite a lot with you when you're explaining all of this, because I'm hearing words I've never heard before, which is quite cool. So thank you very much. I really appreciate it. And if anyone wants to get hold of anyone at Genesis, please tell me, how do they get hold of you guys?
Well, you can contact us on our telephone number, which is 012-367-4378, or you can send an email to reception@genesisart.co.za, or you can go onto our website and make an inquiry, which is www.genesisart.co.za.
Thank you very much. And I know your office is looking quite nice right now. I saw the nice update on your website. So that's nice. I can't wait to see it in person. But thank you again, and we hope to chat to you again soon. Thanks, Neville.
It's a pleasure. Thank you for having me. And on behalf of Genesis, we also want to wish each and every one of you God's best, richest, and choicest blessings.
Thanks, Neville. Bye.
Take care. Bye.